Taking a sample of your own biology is as simple as gently scraping the inside of your cheek with an ice cream spoon. But under a microscope, these cells, and even those from an onion peel, are almost invisible. They are essentially transparent. Without intervention, the internal machinery driving these cells remains hidden. To make these structures visible, biologists introduced chemical stains binding to specific parts of the cell. We rely on these chemical reactions to distinguish individual structures, turning a clear sample into a detailed map of biological activity. Common dyes like iodine for plant tissues or methylene blue for human cells allow us to see what was previously invisible. This diagram shows how different regions of the cell respond to stains. Because each part has a unique chemical composition, they absorb dye at different rates. During this process, a specific dot-like structure near the center absorbs the most dye, appearing much darker. This prominent, darkly stained feature is the nucleus. This structure coordinates the cell's chemical activities and determines how it will develop as it matures. Zooming in on the surface of the nucleus reveals its protective outer boundary. This is the nuclear membrane, and it consists of two distinct layers. The membrane is not a solid wall. It is covered in small pores. These pores act as gateways, regulating the movement of materials between the interior of the nucleus and the surrounding cytoplasm. This creates a secure environment, allowing the nucleus to protect its contents while remaining in constant, controlled communication with the rest of the cell. Inside this protected space, we find DNA molecules, the instructions for building and organizing the entire organism. Most of the time, while the cell is carrying out its daily functions, this DNA takes on a very specific physical form. This is chromatin. It appears as an entangled mass of thread-like structures filling the nuclear space. However, when the cell begins the process of reproduction and prepares to divide, this loose arrangement changes. The chromatin organizes into distinct, rod-shaped structures that are much easier to move and copy without error. We call these organized rods chromosomes. By switching between these two physical states, the cell can effectively manage its daily survival while ensuring its genetic information is perfectly preserved for the next generation. This diagram compares the cells we've been looking at to a common bacterium. The internal layout is strikingly different. Notice that the bacterial cell on the right lacks a nuclear membrane. There is no physical wall separating its instructions from the rest of the cell. In these simpler organisms, the genetic material sits in an undefined region called a nucleoid. These simple organisms are known as prokaryotes, and they represent the most primitive forms of life found on Earth. Organisms with a defined membrane-bound nucleus, like the cheek and onion cells we observed, are called eukaryotes. This single structural development, the ability to house genetic code within a protected membrane, is the defining feature that allowed for the evolution of complex multicellular life.